Hi, it's Dr. Byron. I love me some histology. We're going to talk about extracellular matrix. By the end of the session, I want you to be able to discuss the structure and functions of ground substance in extracellular matrix. The extracellular matrix provides a mechanical and structural support. Like this jello mold here, we have the fruit, which can mimic the cells in the extracellular matrix or the fibers in the extracellular matrix, but the gelatin or the jello is the actual extracellular matrix. There's stuff in there that's very important. They anchor cells within tissue, provides tensile strength, as well as a pathway for cell migration. They can also be a biochemical barrier as well as influence extracellular communication. Now, back in the day in Foundations of Medicine, Dr. Romana had covered with you all an osmosis video on extracellular matrix. This video went through the integrin and cadherin proteins, which are adhesive proteins that anchor and link cells together or structures together. You can have structural and fibrous proteins, which Dr. Romana had covered in FOM, and I also covered the collagen and elastin fibers, as well as the reticular fibers in the connective tissue lecture. In this particular lecture, we're going to focus on the proteoglycans and other substances within this extracellular matrix. You may come across the term ground substance, and it's comprised of proteoglycans, glycosaminoglycans, and multi-adhesive glycoproteins such as laminin, fibronectin, and osteopontin. With proteoglycans, they're comprised of a core protein that is linked to hyaluronan, which you see here as these little green fibers. They are also attached with different types of GAGs, glycosaminoglycans, which are disaccharide units comprised of, again, N-acetylgalactosamine or N-acetylglucosamine, plus uronic acid. Now, do not feel threatened here. You don't need to memorize this. Just appreciate that GAGs are disaccharide units, and they can be very complex in their structure. The role of glycosaminoglycans is mainly for the key physical property of ground substance. They arise from a post-translational modification. So they're made, and then they are modified after they're made. I call this a hydrated gel effect. Basically, just think of a gelatin or a gel insole, right? Why is it gelatinous? There's a lot of water composition associated with it. These proteins have highly negative charge. They have significant osmotic pressure and high swelling capacity. So again, it also resists compression. So when you think of that gel insole, right, it gives a little bit, but it doesn't fully deflate. So that's the resistance to compression. And you have this kind of gelatinous structure, which has a nice structure that gives it a very gelatinous type of appearance because it's holding on to that water. Now, don't feel threatened with this table here. I just want to give you some examples. I don't want you memorizing it, but you do need to appreciate that under the category of glycosaminoglycans, you have your hyaluronan, which we'll talk about in a few minutes. There you have other sulfates, chondroitin, dermacan, keratan, heparan sulfates, as well as, again, heparin. Now, I don't want you to memorize the composition here, but just appreciate that GAGs have a disaccharide composition. They can be seen in various parts of the body, not only extracellular matrix, but they can be seen in cartilage and other connective tissue in the body. We'll talk more about hyaluronan in a second. Now, what do you need to know from the functions, right? But these are key glycosaminoglycans that give the extracellular matrix its support. Now, with hyaluronan, it's a specific type of GAG, which has a different type of structure. It's long, rigid molecule comprised of carbohydrate chains. It can bind to the proteoglycan GAG aggregate. Remember that green fibrils that we saw earlier? It can hold a lot of water. It's like a really big gelatinous sponge. And so some of you may have seen at the grocery stores, right, or the drugstores, the hyaluronic acid for skin, right, anti-aging creams, right? It gives the skin a feeling of smoothness, and it gives the skin a little bit more of that swelling to give someone 10-year age young look. So these hyaluronan molecules are synthesized by enzymes on the cell surface, so there's no post-translocational modification seen with GAGs. It functions mainly as shock absorption. It can immobilize molecules within the extracellular matrix, and it can contribute to lubrication and insulation effects as well. Now, multi-adhesive glycoproteins are small proteins that possess binding sites for various ECM proteins. They interact with cell surface receptors. Their function is mainly to stabilize the ECM by binding to various proteins. They can link to cell surface and help modulate cell movement and migration. Multi-adhesive glycoproteins include fibronectin, and there's many different types of fibronectin, but the main commonality is that they contain several binding domains that can bind to various proteins and play a role in cell attachment. Laminin, we discussed this when we discussed basal lamina and basal membrane. It can bind to collagen type 4 and GAGs. Osteopontin is very unique to the ECM of bone. It binds to osteoclasts, which are the large multinucleated cells that break down bone. Class means to break down, and so it can attach the osteoclast onto bone. They can also sequester calcium and promote calcification of the extracellular matrix as well. So just to recap, we've discussed some key elements of ground substance within the extracellular matrix, which include proteoglycans, glycosaminoglycans, hyaluronan, and the multi-adhesive glycoproteins. They have various significant roles within the extracellular matrix, so these are key players and not to be ignored. On that note, please email me or contact me if you have questions or need clarifications, and I will see you at the next lecture. Thank you. Bye!